Hello, everyone. I'm Diksha Devkota, a second year medical student here at Nepalese Army Institute of Health Sciences, Nepal. And today I am going to attempt to give a brief overview of the development of heart. So, without further ado, let's get started. Heart is the first functional organ to develop, which begins developing in the middle of the third week of our intrauterine life, and it begins functioning at the end of the third week of intrauterine life. And our heart is mesodermal in origin, and it develops from the primitive heart tube, which we will discuss later. What are the general concepts that we will require while studying the development of heart? So, firstly, we need to know that intraembryonic mesoderm, it has three parts: the paraxial, intermediate, and lateral plate mesoderm. So, this black is the paraxial mesoderm. This is the intermediate mesoderm, and this is the somatic mesoderm, somatic layer of the somatic layer of the lateral plate mesoderm, and this is the splanchnic layer of the lateral plate mesoderm. And our heart, it develops from this splanchnic layer of the lateral plate mesoderm. So this is the epiblast, and this is the endoderm, ectoderm, and the endoderm, and middle one is the mesoderm. So the progenitor heart cells from the epiblast, they migrate into the mesoderm, into the splanchnic mesoderm layer, and here they are called the primary heart field. So let's see in this figure, the progenitor heart cells, they migrate through the primitive streak into the cranial portion of the embryo, of this flat embryo, and it forms this inverted U-shaped structure, or the horseshoe-shaped structure, which is named as the primary heart field or the cardiogenic area. This primary heart field, it forms from the days 16 to 18, and it forms the major part of the heart. That is, it forms both the atria, and the left ventricle, and most part of the right ventricle as well. And the remaining part, that is the some part of the right ventricle and the outflow tract of both the ventricles, it's derived from the secondary heart field, which develops a little later than the primary heart field, that is from days 20 to 21. And the function of the secondary heart field is to elongate the heart. That means, as it forms the outflow tract, so it elongates the heart, and if there is any defect in the secondary heart field, that will result in the shortening of the outflow tract, so that will lead to outflow tract defects. So now let's talk about so the endoderm that underlies this primary heart field. Endoderm that underlies the primary heart field, this yellow portion, it secretes vascular endothelial growth factor. This vascular endothelial growth factor, secreted by the endoderm underlying the primary heart field, it tells the primary heart field cells to self-organize into two endothelial heart tubes. These two endothelial heart tubes, so two endothelial heart tubes have now formed from the primary heart field. Now, during the lateral folding of embryo, there are certain changes that the tube undergoes. So, firstly, the two heart tubes, they fuse in craniocaudal direction. So, the during the lateral folding, what happens is that till now our embryo was flat. Okay, so now the flat embryo, it becomes cylindrical due to the lateral folding, and during this lateral folding, two endothelial heart tubes also fuse to form a single endothelial heart tube, and they fuse in craniocaudal direction. So this is the first thing that happens during the lateral folding. And other things what happen is, the two pericardial cavities, they also fuse to form a single pericardial cavity. The two dorsal aorta, they fuse to form a single aortic sac, and the two vitelline veins, they fuse to form a single sinus venosus. And now let's talk about the craniocaudal folding. In the lateral folding, our flat embryo has become cylindrical. Now our cylindrical embryo will become curved down. It will curve down, and during this craniocaudal folding, which happens in the fourth week, beginning of the fourth week, what happens to the heart? What happens to the heart is that heart, which was in the cranial portion of our embryo till now, it descends into the thorax. Now our heart is in the thorax, and the heart also develops different saculations or dilatations, as we can see in this figure, which we will discuss later as well. Now, for now, let's discuss about the development of the heart wall and pericardium. Now, before the craniocaudal folding of our embryo, our endothelial heart tube, the primitive heart tube, it, it used to be located below this pericardial cavity. But now, after the craniocaudal folding, they undergo 180-degree rotation so that the endothelial heart tube it comes to lie in the roof of our pericardial cavity, and the mesoderm overlying this pericardial cavity, it thickens and forms the myoepicardial mantle. So now slowly along with the development, this myoepicardial mantle, it completely encloses our endothelial heart tube or the primitive heart tube, so that our heart so that different layers of heart is different so that the different layers of our heart wall is formed, and also the pericardium is also formed. So this the endothelial lining itself of the primitive heart tube, it forms the endocardium. The myoepicardial mantle, this myoepicardial mantle, it forms the myocardium as well as the visceral layer of serous pericardium. As we know that pericardium has two main layers, serous pericardium and fibrous pericardium. The serous pericardium has further two more layers, that is visceral layer and the parietal layer. And the visceral layer of the serous pericardium, it's formed by the myoepicardial mantle. So the myoepicardial mantle forms the myocardium and the visceral layer of serous pericardium. And the uh, parietal layer of the serous pericardium and the outermost fibrous pericardium. Outermost fibrous pericardium, these two structures, these two layers are formed by the somatic mesoderm of the lateral plate mesoderm. So somatic mesoderm forms the parietal layer of serous pericardium, and outermost fibrous pericardium. I hope that's clear. Now, let's talk about the dilatations that have appeared in our heart tube after the craniocaudal folding. So, as we discussed here, our heart has developed dilatations. So, what are those dilatations? Those are from cranial end to caudal end. Truncus arteriosus, bulbus cordis, primitive ventricle, primitive atrium, and the sinus venosus. So truncus arteriosus is included in the bulbus cordis in some books, but it doesn't make any difference. So truncus arteriosus, it's the arterial end arterial end or the outflow end of our primitive heart tube, whereas the sinus venosus, it's the venous end or the inflow tract of our primitive heart tube. So this is the same thing. Now, the arterial end. This is the arterial end. This is the bulbus cordis, truncus arteriosus. So the truncus arteriosus, it gives rise to the aortic sac, and this aortic sac joins with the first pharyngeal arch artery. This first pharyngeal arch artery, it it travels laterally to the foregut, and joins with the dorsal aorta. So truncus arteriosus, it aortic sac, which gives rise to the first pharyngeal arch artery, which further joins with the dorsal aorta, right and left dorsal aorta. And in the venous end, we have sinus venosus, and sinus venosus has two horns, that is the left horn and the right horn, and each horn is formed by the union of three veins. So, what are those three veins? First one is the common cardinal vein, then the umbilical vein, and the vitelline or the omphalomesenteric vein. So, this is the venous end, and this is the arterial end of our primitive heart tube. Now, let's talk about its derivatives. Now, the bulbus cordis, the distal one third is called the truncus arteriosus, or truncus arteriosus itself. It gives rise to the ascending aorta and the pulmonary trunk, whereas the middle one third, which is the conus cordis, it gives rise to the outflow tract of the right and left ventricles. And the proximal one third, it forms the rough part of the rough trabeculated part of the right ventricle. Actually, the right ventricle, some part of the right ventricle is formed by the proximal one third of bulbous cordis, and some part is formed by the primitive ventricle as well. So the bulbous cordis and the ventricle, primitive ventricle, they are separated from each other by the bulboventricular sulcus. But this sulcus doesn't appear already; it appears only after the cardiac looping. So in this primitive heart, there is only a ridge, bulboventricular ridge. So the primitive ventricle, it gives rise to the rough part of the left ventricle and some part of the right ventricle as well. And the ventricle and atrium, they are connected by the atrioventricular canal. And the atrium, it is partitioned. Primitive atrium, it's partitioned to form right and left atrium, only the rough part. And the sinus venosus, the sinus venosus has body and two horns. um, the right horn is observed into the right atrium, and it forms the smooth walled part of the right atrium, whereas the left horn, it regresses, and the remnant structure, it only forms the coronary sinus and the oblique vein of the left atrium. So rough part of both the atrium is formed by the primitive atrium, and the rough part of the right atrium is formed smooth part of the right atrium is formed by the sinus venosus, right horn of sinus venosus, whereas the smooth part of the left atrium is formed by the pulmonary veins. So we need to remember that. And the different veins unite to form the horns of the sinus venosus. So the common cardinal vein, it forms a part of the superior vena cava. Right umbilical vein, it eventually disappears, whereas the right vitelline vein, it forms the part of the inferior vena cava. So these are the derivatives. Now let's go towards the formation of the cardiac loop. So cardiac loop, it cardiac loop, the looping of the heart, primitive heart, begins on day 23. So this cranial portion, it bends ventrally, caudally, and to the right. So ventrally, caudally, and to the right, like this. And the caudal part, it moves, it shifts dorsally, cranially, and to the left. In this way, the cardiac loop is formed, and the bending, it occurs due to some cell shape changes. And by day 28, the cardiac loop is formed. This will be more clear in this figure. So previously, our heart tube was suspended in the pericardial sac with the help of dorsal mesocardium. Dorsal mesocardium suspends this heart tube in the pericardial sac, but pericardial sac, and but along with the development, transverse pericardial sinus appears here. And finally, the whole dorsal mesocardium, it disappears. Dorsal mesocardium gradually disappears, and this bulboventricular loop becomes U-shaped, and the bulboventricular sulcus appears here. And uh, the caudal part, which was previously embedded here in the septum transversum, it also becomes free, and it enters the pericardial sac. Now, after all these phenomena, our cardiac loop becomes S-shaped. Okay, so this is it. Now, along with our S-shaped cardiac loop, it it becomes the ventricles grow more and more, and it moves the ventricles move towards the right side, I mean towards the left side, and it covers the primitive atrium. And by the end of the week 4, our cardiac loop almost looks like an adult heart, but only externally. Internal partition is still left. So this, by day 28 or week 4, end of week 4, externally it resembles an adult heart. Now let's talk about the internal partition of heart. So primitive heart tube, it has single lumen, as it is not partitioned. So it needs to be divided internally with the help of 4 septa. The atrioventricular septum, the interatrial septum, interventricular septum, and the aorticopulmonary septum. Now, first, let's talk about the atrioventricular septum. So still now, our heart has a single AV canal. There are no right and left. So the walls of the this is the antero-posterior view, and the walls of our heart, they grow. The endocardial cells of the walls, they grow towards each other, grow, and they fuse to form a single endocardial cushion. They fuse to form a single endocardial cushion, and this endocardial cushion, it divides our heart, our AV canal into right and left, left AV canal and right AV canal. So this endocardial cushion is called atrioventricular septum or septum intermedium. So this is the septum intermedium, and eventually from the ventricular side, the endocardial cells proliferate and form the valves. And after the formation of valves and two separate AV canals, our atria and ventricles are finally separated. I hope you are understanding what I'm saying. Now let's talk about the interatrial septum. So it's a little bit complicated, but it's easy in this. The osmosis has made it easy, but so the wall the wall of the heart, the uppermost wall of the heart, it gives rise to a septum. The atrial wall, it gives rise to a septum, which descends down and leaves a small opening. This septum is called septum primum, and the small opening that is left for a short amount of time is called the ostium primum. And the septum the septum primum, it eventually closes the ostium primum, but still I don't know why it's it's written. But the septum primum appears, it descends down, leaving a small opening, which is the ostium primum, and it eventually closes the ostium primum as well. Then another opening appears here in the septum primum itself, which is called the ostium secundum or the second opening. This ostium secundum appears, and in the attempt of closing this ostium secundum, another septum appears from the right side of the septum primum, which is the septum secundum. And this septum secundum also leaves a small opening here, which is called the foramen ovale. So our septum primum is thin and mobile, whereas our septum secundum, it's thick and immobile. So blood can, since in the fetal life, the blood pressure here in the right atrium is more. So the increased blood pressure will push this increased blood pressure will push the blood from through the foramen ovale, and it will push this septum primum, so that the blood travels from the right atrium to the left atrium. But after the birth, the left atrium also starts receiving the blood from the lungs. So the pressure in the left atrium exceeds the blood pressure in the right atrium, such that this two septums, they start opposing each other, and through the opposition they fuse. That means that interatrial septum is formed. The blood pressure of the left atrium, it exceeds the blood pressure of the right atrium, and these two septums fuse and form the these two septum fuse, closing the foramen ovale and forming the interatrial septum, which will prevent the flow of blood from left to the right atrium. So that is it for the interatrial septum, which divides the atria into right and left. Now let's talk about the interventricular septum. It's not quite as complex as the interatrial septum. So muscular ridge arises from the floor of the ventricle, and the membranous ridge, it arises from the endocardial cushion of the AV septum, this AV septum. So they come in close proximity, they fuse and form the interventricular septum, which divides to two ventricles. But Indian books have made it complex and added another thing, that is the bulbar part, the membranous part, and the muscular part. So we can refer the bulbar and the membranous part as the membranous region. So that will be okay, I guess. So in this way, our interventricular septum is also formed. Now let's talk about the aorticopulmonary septum, which is a spiral septum, and it divides our truncus arteriosus into the aortic channel and the pulmonary channel. So in the truncus arteriosus, we need to know that there are two swellings: the right superior truncus swelling, and the left inferior truncus swelling. These two swellings, uh, this right superior truncus swelling, it moves distally and to the left, and the left inferior truncus swelling, it grows distally and to the left. That means they grow opposite to each other, so they form a spiral trajectory and wrap around each other. So and eventually at last they fuse, and after the fusion, the aorticopulmonary septum is finally formed, and it divides our truncus arteriosus into aortic channel and pulmonary channel. And there are also two more swellings here: right dorsal conus swelling and left ventral conus swelling. These two swellings are present in the conus cordis region of our bulbus cordis. These two swellings also, this right dorsal swelling moves dorsally, moves distally, and this left ventral conus swelling moves also move distally, and they also wrap around each other. They also wrap around each other and unite with this truncus septum. They unite with the truncus septum, and finally they divide when they fuse. When these two swellings, these two conus swelling fuse, they divide the conus cordis into outflow tract of the right ventricle and the outflow tract of the left ventricle. So the truncus swelling, truncal swelling, they undergo a spiral trajectory and finally form the aorticopulmonary septum, whereas the two conus swelling, they they fuse finally after uniting, after growing distally. They fuse and form a septum which divides the conus cordis into the outflow tract of the right ventricle and the outflow tract of the left ventricle. I hope it is making sense. um, Now, the development of the venous system, I have already explained this, but still, let's go through it one more time. The sinus venosus, it receives the blood from the vitaline, umbilical, and common cardinal vein. But after but soon the sinus venosus becomes asymmetric because the left horn regresses and the right horn, it enlarges. So the right horn, it enlarges to become the smooth walled part of the right atrium, as I already discussed. But the left horn, it shrinks and persists only as the coronary sinus and the oblique vein of the left atrium, whereas the right horn, it enlarges and forms the becomes the smooth part of the right atrium, and forms the opening for the superior and the inferior vena cavas. Then conducting system. So conducting system are also the same myocardial cell, but they are a special group of myocardial cells which organize and form pacemaker centers. So the cells in the wall of the sinus venosus, wall of the sinus venosus in the right atrium, it forms the sinoatrial node. The cells in the atrioventricular septum, AV septum, they form the atrioventricular node, and the cells in the interventricular septum form the which is its location. um, and the rest of the ventricular myocardium, the cells of the rest of the ventricular myocardium, they form Purkinje fibers. So that's it. I hope this is not the case. I hope that you did retain something. Thank you for watching. And the references for my presentation and my understanding, it's from Langman Embryology, from Osmosis, and from the textbook of Embryology by Vishram Singh and textbook of Embryology by IB Singh. So thank you for watching. I hope it made sense. Thank you.